Thank you, Christine, and welcome to Prime Innovation Showcase. I'm Milica Vukmirovic, Director of External Partnerships and Programs, and I'll be moderating today's Prime Innovation Showcase. Before I announce company presentations, I would like to briefly acknowledge that Prime PIs and their teams are very translational and very entrepreneurial. There are more than 20 startup companies co-founded by Prime PIs and more than 250 capital dollars raised, so quite impressive. And within Prime team in the last year, we actually created more than 35 academic and industry collaborations. And with this, we would like to continue this positive trend, and we would like to create more. Today's we have 3 companies presenting at our innovation showcase. So our first presenter is Mohammad Ali Amini, who is the CEO of Nanology Labs, and he co-founded the Nanology Labs with Shirley, professor from Leslie Then Faculty of Pharmacy. Our second presenter is Seray Cicek, who is the CEO of LSK, and she co-founded LSK with Keith Pardee, assistant professor of Leslie Then Faculty of Pharmacy. And our third speaker is Somasekar Seshagiri, CEO of AntlerA, and he co-founded that company with 2 professors, Sachdev Sidhu from Department of Molecular Genetics, and Stephane, who is director of Donnelly Center for Cellular and Biomolecular Research. So our first speaker is Mohammad. And he will be talking about Nanology Labs. Nanology Lab is developing smart cancer, nanomedicine, and drug delivery platforms. So please, Mohammad.
Thanks so much, Malika, and thanks for the invitation. Allow me to share the screen and walk through the slides together, and that will be much more easier. Okay. Okay, hello everyone. As you introduced earlier, my name is Mohammad. I'm the CEO and co-founder at Nanology Lab. So what we do at Nanology Lab is developing advanced nanomedicine to ultimately offer curative treatment to cancer patients and send them home back to their loved ones. As you might know, technically Nanology Lab is one of the University of Toronto spin-off company. It was technically formed based on 2 strong intellectual property developed at Dr. Shirley Wu's lab at Faculty of Pharmacy at U of T. Shirley was actually my PhD supervisor, and the technology we're currently working on was part of, a major portion of that was part of my PhD thesis. Early discussions around building the company goes back into 2017, early 2018, where we started building the the business plan around the technology and forming Nanology Lab as it is today. And actually, we moved the company right across the street to JLab's facility, which is a very nice sort of ecosystem for entrepreneurs and any sort of commercialization activities. What we are doing as our lead program technically at Nanology Lab is around building and developing TMX, which is a tumor modulating therapeutic asset we are trying to overcome with the challenges, treatment resistant challenges we are faced in clinic in treating solid tumors. So as you know, in treating tumor, technically solid tumors, we have a very key barrier, which is known as lack of oxygen in the tumor or tumor hypoxia. Let me go to laser view over here, if I can find it out. I'm not sure if this is okay. So I'm just going to quickly introduce what is tumor hypoxia and what is the challenge we have in treating treatment-resistant solid tumors. So under a lack of oxygen, deeper layers in the tumor tissue, there's lower levels of oxygen, which is known as hypoxia. And under this hypoxic condition, cancer cells start changing their metabolic and signaling pathway, making the the cancer cells generating more reactive oxygen species such as hydrogen peroxide, overproducing some of the very invasive genes such as HIF-1 alpha, and also they start producing more protons, making the tumor environment very acidic. All in all, these factors lead into having an environment which is highly treatment resistant, immunosuppressive, and angiogenic. About 10 million of the patients, cancer patients all around the world being diagnosed with hypoxic solid tumors, and half of which patients are being actually losing their lives of it, sadly. So as you can imagine, there's a huge unmet need and a huge barrier in clinic to treat solid hypoxic tumors. At the Nanology lab, we came up with a very advanced and practical solution to treat these solid tumor tissues to overcome technically hypoxia. TMX is a sort of the next generation anti-hypoxic or anti-hypoxia agent that is in form of IV formulation. Upon IV administration, as you can see, they actually start accumulating inside the tumor tissue, and they react with cancer cells byproduct as shortly touched on ROS, such as hydrogen peroxide and protons. And upon this reaction, they start producing the element of oxygen. By producing oxygen inside the tumor tissue, we are actually initiating a series of actions in the tumor, which leads into not only improving the tumor oxygenation and neutralizing the pH, but also causing more damages to DNA of the cancer cells along with other therapies, inhibiting some of the very aggressive genes such as HIF-1 alpha, VEGF, which leads into inhibiting the angiogenesis, and also lowering down the immunosuppressive factors, and which leads into boosting anti-tumor immunity. Here's some of the very sort of snapshot of one of our preclinical studies, just to show how it works and what is the sort of treatment indication here. Here is the monotherapy and combination therapy with TMX. As you can see in a xenograft prostate tumor model, the monotherapy with TMX leads into improve the life expectancy by twofold. When it combines with radiation therapy, it maximizes the treatment outcome, the efficacy of the radiation therapy, which leads into up to 40% curative treatment, which means complete tumor eradication, and also offers 5-fold improvement in life expectancy, which is phenomenal compared to other tumor modulating or hypoxia activated prodrugs out there in clinic or in pipeline. So all in all, this product offers a series of advantages compared to the commercially available or in pipeline products. First of all, it's a direct approach in overcoming hypoxia by generating element of oxygen locally inside the tumor. It works independent to tumor heterogeneity, unlike many biologicals out there, and can address and target all types of solid tumors. And it has a very nice streamlined and scaled manufacturing process. We can make liters of this formulation with very high batch-to-batch consistency, which is one of our recent milestones accomplished. We have covered the technology with a series of patents. Some of them has already been granted some past PCT, and as you can see, the science has been well established and validated by publishing in top-tier journals. What we're targeting, our lead indication generally is to focus on patients with prostate cancer. And we're technically, as the lead indication, we're targeting patients with intermediate to high risk patients with prostate cancer, which engulf about 30% of the total population of cancers in this category. Sadly, the median survival rate for this portion of the patient, subset of patients, is it's up to 30 months. And total estimated annual obtainable market is about $800 million. We've been working on this technology since 2014 on the discovery zone, and we moved it through a lot of grants into collecting and de-risking the technology at the preclinical setting with running a lot of efficacy studies. And we actually recently closed our seed financing, which is supporting all the IND enabling studying and positioning Nanology Lab to the next key value inflection point. And one of the key objectives, the next key objectives for the company, and that is launching the lead program TMX into phase 1 clinical trial, for which we are currently raising the Series A. Our lead indication is to treat patients with prostate cancer using TMX IV administration along with radiation therapy in the phase 1, observing those low toxicity study to determine the maximum tolerated dose of TMX in a phase 1 setting. And thanks for your time today, and happy to answer any questions. I would like to thank our supporters, which was led actually by FACIT, University of Toronto, a lot of local institution and investors in Canada. Thanks.
Thank you, Mohammad, for sharing this exciting data from Nanology Labs.
No worries.
And it's really exciting time actually for Nanology Labs, like to be kind of in AND enabling, level, kind of thinking about going into phase 1. So I would really encourage people to post their questions in the chat box. For the sake of time, I would like to invite our second speaker to present LSK company. So Seray, please go ahead.
Good afternoon, everyone. I'm Seray, Cicek, CEO and co-founder of LSK Technologies. And at LSK, we bring lab-grade diagnostic testing without the lab to point of need. And today I'm going to share with you our journey so far. But before I begin, I would like to introduce my co-founders. Our company is co-founded by Livia Guo, who's our COO, and Dr. Pardee, who's our CSO. Livia and I completed our master's in pharmaceutical sciences under Dr. Pardee's supervision at University of Toronto. My first part of the talk will focus on how we got started as a startup from the lab and what motivated us to pursue. Our journey started in 2015-2016 Zika outbreak, when Zika was an endemic in a local outbreak in Brazil, Colombia, and Ecuador. Zika is a vector-borne disease, and it is transmitted by mosquitoes. Zika, similar to what we're experiencing today, was a known virus since 1950s, but it wasn't until late 2015 that its link to microcephaly was discovered. It was hard to diagnose. It had common signs and symptoms. So a laboratory diagnosis was needed. And 4,000 microcephaly cases occurred in 2015-2016 outbreak, highlighting the urgent need for decentralized diagnostics and affordable diagnostics. To answer to this call, Pardee and Green Labs developed an affordable paper-based diagnostic that changed color if the sample was infected from yellow to purple. We collaborated with labs in Ecuador, Colombia, and Brazil for field studies. However, there was a problem. Our collaborators did not have the devices that we were needed. Our reactions were cost-effective at 384 well plate size, and our collaborators had 96 well plate readers. Because of this dilemma, we had a problem, and this inspired us to create a solution. So our solution is Plum. It's an affordable and portable plate reader. Unlike traditional devices that focus on hardware, we use software to quantify reactions, and we do so with the help of a camera. Simply, we take standard lab plates, well plates, we take pictures of that, find the walls of interest, quantify them, and output a graph, as you see on the right-hand side, that can differentiate between a negative and positive result. Our devices are connected to cloud, so results can be easily shared with collaborators worldwide. After checking the box that we can detect paper-based Zika diagnostics, we moved on to expand the platform to other organisms and molecules. Our platform can detect colorimetric and fluorescent reactions. We can process standard protocols such as isothermal amplifications and ELISAs. All of this combined enable us to detect different targets, antibodies, antigens, nucleic acids, and small molecules. We had a unique opportunity to create 3 prototypes and deploy them in Ecuador, Brazil, and Colombia. Livia and I actually traveled to Ecuador and Brazil and also supported the clinical tests. In Brazil, we obtained 98.5% accuracy in a 268 patient trial for detecting the Zika virus. This, combined with seeing the positive feedback from our users, made us explore commercialization routes for our product. In January 2020, Livia and I graduated and started working on our company. But before we jump into our journey, we focused on raising funds and validating the market. So Velocity Pitch competition was a pivotal win for us, which we received our first big check size that allowed us to relocate to Kitchener, start our lab space and office space. But in addition to that, we moved on to raise $500,000 in funding, and we received recognition from various competitions that are listed here. We are a Y Combinator-backed startup, and we have local support from H2I Incubator, as well as UTest and TIAP. Now we are working full-time on our company. So here's what we do today. As LSK Technologies, our focus is to decentralize diagnostic testing, and we have 2 product lines. First one is Plum, which is a pop-up lab capacity, and second one is Flux, which is more so a decentralized diagnostic device. And for Plum, it's an affordable plate reader that utilizes camera vision to quantify optical reactions. It accepts standard plates. In addition to that, we have also developed a room temperature stable all-in-one RT-LAMP kit that can detect SARS-CoV-2 RNA. When used with our device, we can quantify these reactions and provide graphical output, as you see on the right-hand side. And our devices and the kits are available for research and education use. Our highlight is that we are 5 times more affordable. There is no cold chain storage required for our kit, and our device is adaptable to multiple optical reactions. We have equivalent performance to qPCR, and we can also detect RNA without extraction as well. Our second product line, it focuses on point of care diagnostics for pharmacies and is currently in development. We realize that there is a huge demand for pharmacies, and they're currently overburdened by the diagnostic demand. Pharmacies also remain to be the most accessible healthcare unit, and our solution allows them to conduct their workflow seamlessly. When a patient comes in, by scanning QR codes, they can be entered to our system. Sample is conducted in our sample cup and cartridge, and within 6 minutes, within 30 minutes, 6 customers can be analyzed per device. At the end, our results can be forwarded to cloud and shared with third-party apps. Our Flux unit is also flexible in terms of sample collection and can detect different targets such as nucleic acids, antibodies, and biomarkers. We have conducted preliminary testing for COVID-19 RNA detection and antibody detection and obtained high accuracy. In addition to that, we can also detect multiple targets within one cartridge, such as flu and COVID, and go up to 12 targets per cartridge. These are room temperature stable and enable end-to-end quality tracing through QR codes. Our ultimate goal is to collaborate with assay developers so that they validate their tests in our Plum lab unit that can be later onboarded into our point of care Flux unit. With this, we enable to unlock more tests for patients. Today, we are happy to work with academia, industry, and venture capital partners. In terms of academia, our devices are available for research and education sales. You can reach us with the email and website provided down at the below. And also, we're open to partnerships for grant applications and research collaborations as well. In terms of the industry, we are happy to partner with strategic groups who are willing to help us with their expertise in regulatory and distribution. And we are raising our 2 million seed round, so we're happy to chat with venture capital partners. Thank you for listening. I know that there's no more questions, so I hope you enjoy the rest of the event. Thank you for having me.
Thank you, Seray, for amazing presentation and very inspiring journey from Zika to actually SARS-CoV and to actually have an instrument in the time of need, in the middle of pandemic. I mean, truly impressive. Thank you once more for your presentation. And we are going into our third presenter and the company AntlerA. Company is actually aimed to develop novel protein therapeutics to modulate Wnt signaling and control tissue repair. And presenter is Somasekar Seshagiri.
Apologies, I was just trying to unmute. Okay, thank you, Malika and Christine. I'm going to try and tell you a journey on a protein engineering meeting biology, a combination of which is produce some very powerful platform, but also opportunity for many therapeutics. So AntlerA was founded in Toronto by Dev, Sidhu, and Stephane almost 3 years ago. And the logo you see here on the top actually comes from Canadian caribou, you know, in your coin in the back. And the reason for it is this antlers fall off and regrow during the sort of, you know, past fall. And underneath is the Wnt signaling pathway that's very critical. So as we named the company, we try to make it as Canadian as possible. And so we decided to add an A at the end. And of course, fundamental, all the jokes aside, is a very conserved biological pathway that's very important in the context of regeneration. And today, regenerative medicine, typically, if you hear about it, it's all stem cell transplants and so forth, but not protein therapeutics that can facilitate stem cell renewal and homeostasis. And what that does is makes one drug that's suitable for across patient population. So it is a picture of a little tiny worm. And you can cut it in 1,000 pieces and you can put it in the right nutrient media. It will regrow. This little flatworm will reproduce its primitive brain and eyes remarkably well over and over. And it's a bag of stem cells under the right conditions. It can just make a whole copy of itself. You move up in evolution, you look at this axolotl. So it's a very cute little salamander with remarkable regenerative ability. On the right here is a panel. If you amputate one of its limbs, it will completely regrow and you cannot tell that there was an amputation. And you can do it many times, not only the limbs, but its vertebrae, its gills, lungs, and so forth. And this has become a model system study, actually, spinal cord injury and regeneration. And again, underneath all of this is this critical pathway, the Wnt pathway. And as you go up in evolution, we have lost the ability to completely regenerate full organs, save the liver. But we do have a lot of stem cells. And the idea here is not to regenerate organs completely, but to bolster the stem cells and allow them to renew so degenerative diseases can be addressed. You know, in an adult human, every 4 days you replace all the intestinal cells, and so do we, all our skin cells. And for all of this, there are stem cells within these tissue compartments, and they all rely on the Wnt signaling pathway, which is conserved across evolution. So what is this Wnt pathway? There are 19 Wnt ligands, and there are 2 receptors to which they bind, one called frizzled and another one called LRP. There are 10 frizzles in our genome, and you go down evolution, also this is conserved. And you got 2 LRPs that are being used. And downstream of it is a signaling circuit that gets turned on and does a number of things. Of course, people have talked about using this Wnt ligand, which originally, incidentally, was cloned as a ligand promoting cancer because it's just supported uncontrolled cell growth, but in the context of other mutations, not by itself. But still, people have been talking about this Wnt ligand as a hormone, just like insulin. You guys know in Toronto there, the 100th year of insulin. This Wnt ligand could have the potential for doing something very important in supporting stem cells and their renewal and tissue homeostasis, and particularly in degenerative diseases. But unfortunately, Wnt ligand is very hard to make. The nature of the protein is such that it has many cysteines, and on top of it, it has a lipid that's attached to a conserved serine. So this makes it highly hydrophobic and impossible to make a protein that can be used as a drug. And on top of it, one Wnt can actually bind many frizzles, so they can activate many frizzles. And so the two colleagues here, Dev Sidhu and Stephane, Dev, one of the world's best protein engineers, and Stephane, the eminent expert in biology, got together and tried to say, hey, can we use antibody engineering to actually turn on the pathway? And out of that came this molecule, which is a bispecific antibody, a tetravalent antibody. On one end, it hugs the frizzle receptor, the other end, the LRP receptors. There are many other constructs in the field, like bispecific antibodies, where you'll have one antibody binding, one arm binding to frizzle, another one binding to LRP. Those don't activate the pathway. This tetravalent configuration was very critical for turning on the pathway, and that's what you see here in a reporter assay. And this was all published in about late 2019 in eLife. And this construct, I just want to kind of point out, is quite modular. When I started, I said Wints are non-specific, they're promiscuous. By adding an antibody at this end, on one end, we can make it very specific. If I add a frizzle-1 antibody, it'll turn on the frizzle-1 receptor. If I add an antibody that's LRP specific, it's going to bind only LRP. And I can switch that to any antibody in the frizzle series and turn on that particular receptor. So it's actually quite modular and programmable. And the way we were able to do this is because we have antibodies against every frizzle, the 10 frizzles that I told you. We have antibodies against the LRPs as well, the 5 and 6. So you put that together, you end up with a library of molecules. This is extremely powerful set of molecules. These are probes that were previously impossible to study this pathway. Now we have these molecules. What you see here at this end and terminal end is frizzle-1 antibody. This is a frizzle-1 agonist. All I need to do is swap this out of the frizzle-2 antibody. It becomes a frizzle-2 agonist. I move from left to right. As I replace each of these, I get varying specificity. And this exquisite specificity also allows me to improve safety in vivo in terms of patients and also turn on the pathway on demand. And the opportunity for regenerative therapy with these types of molecules is enormous in a number of organ areas. But we decided to focus on a couple of areas, given that you can't do everything all at the same time. So we have molecules that are now advancing in the context of eye, another one in the context of IBD. On the eye molecule, it's closer, more advanced. And I'll show you a couple of pieces of data to give you a sense of what I mean by all of this. So this is a fundus image of an eye, and these days you can look in the eye and you can, literally speaking, but also, figuratively looking into the eye, you can actually say a lot of things, looking at the blood vessels, which you see here. And these blood vessels are privileged in the sense, eye is a privileged site, even though it's a neural tissue. If you inject a dye in your vein, dye will not extrude into the retina. And these capillaries are so tight. And there's only a couple of other sites in the ear and the brain that have that privilege, whereas the dye would, saturate other organs. The minute this integrity of these blood vessels is lost, you get leakage, you get these aneurysms, you get exudates, and basically you get swelling of the macula. And anti-VEGFs have been used to treat this for the last 15 plus years. There's not been another drug with a different MOA that has come to the market. And also there is a big need because not everybody responds to VEGF. But the biology underneath this blood vessels integrity goes back to the Wnt pathway. There's a fristle for LRP5 receptors. I told you this is one of the fristles. And the molecule, the ligand here is not actually Wnt, but a related TGF-beta family member called Norrin that suffers the same fate in terms of recombinant production. And that actually turns on the same exact pathway downstream. So AntlerA, using this technology, wanted to make what we call a Norrin mimetic. So here is a blood vessel, and you look at the endothelial cells. This is the frizzled LRP pathway. And this signaling is important to keep these two cells together. It's like a cell-cell junction, the cadherins and the occludins and other stuff here. Think of it like a very tight zipper. The minute the signaling is lost, the blood vessel starts leaking. So if you had a drug that can turn on the pathway, you're going to have a lot of benefit and a number of diseases to take it to. So in fact, that's what we did. We made a molecule that can turn on the fristle for LRP5 specifically. This is the Norrin ligand that you can buy in small quantities. So we know it's quite active. Recently, Stephane and colleagues led some of the study along with Dev to publish this work. And in vivo, we were able to show that it actually blocks pathological neovascularization. This is the control. When we inject a molecule in the eye of mouse, basically, you don't see these tufts, and you can actually quantify them. And what we built actually is a very powerful agonist platform, a Wnt mimetic platform. And they have all the ideal properties. We have further evolved this to a drug-like molecule. We are the first-in-class Norrin mimetic that's moving, inching towards the clinic. We've developed many other molecules for treating IBD, liver regeneration, bone, even Parkinson's for that matter. So these are all work in progress. All of this, would not have been possible without the seed sort of ideas coming out of University of Toronto, particularly Dev and Stephane. And I just want to thank many colleagues, both on the AntlerA side and also colleagues in Toronto that helped us build a company and do things. So I'm going to stop here and just say that if you are interested in positions, we do have a few on the US side of our labs, particularly on the protein expression and purification. So you can email me on the email address above. And I'm going to stop here. And happy to take questions if there is time. If there isn't, no worries.
Thank you, Sekar, for this very informative presentation. And yes, Wnt signaling is well studied, but still we do not have great modulators. And it seems like that you are onto something here with AntlerA, and I wish you good luck in continuation. I'm not sure if we have any questions, but I know that we are very tight with time. And again, I would like to thank all of our speakers in Prime Innovation Showcase, and to encourage audience to really interact. To encourage audience to interact with them, as we could see, they're looking to partnerships. There are some positions open.